Hello students, welcome to my YouTube channel. In this particular video, I will discuss about the C4 cycle that plants perform in order to fix carbon dioxide in its body. Basically, there are two processes by which plants fix carbon dioxide from the atmosphere into their body. One of them is the C3 cycle and second one is the C4 cycle. C3 cycle is basically a formation of the first stable compound, that is the phosphoglyceric acid, which is mainly made up of 3 carbon molecules, and in case of C4 cycle the first stable compounds that plants will produce is the 4 carbon compound, that is the oxaloacetic acid. Now there are also some basic differences between the C3 process and the C4 process. In case of C4 process or C4 cycle, the fixation of the carbon dioxide mainly occurs in the two different ways or two different locations of the plant cell. One of them is the mesophyll cell chloroplast and the second one is the bundle-sheath chloroplast. But in case of the C3 plants, the fixation of the carbon dioxide only occurs in the mesophyll cell chloroplast. So let's start about the C4 plants and what are the basic characteristics of the C4 plants and what are the basic features of the C4 plants that we can see in the different examples. Now, first of all, in C4 plants, there are, as I have told you earlier, that in case of C4 plants, there are basically two locations where mainly carbon dioxide fixation occurs. One of them is the bundle-sheath chloroplast and second one is the mesophyll cell chloroplast. First of all, the carbon dioxide will be entrapped in the mesophyll cell chloroplast from the atmosphere by an enzyme, that is the carbonic anhydrase. Carbonic anhydrase, which makes carbonic acid by reacting carbon dioxide with water in the mesophyll cell chloroplast. Now this carbonic acid will be broken up into hydrogen ion and bicarbonate ion, and this first bicarbonate ion will be accepted by a particular bicarbonate acceptor which is already present in the mesophyll cell chloroplast, that is the phosphoenol pyruvic acid. Phosphoenol pyruvic acid is a 3 carbon compound. Phosphoenol pyruvic acid, as the name implies, it is the pyruvic acid which contains a phosphate group. That's why it is called the phosphoenol pyruvic acid, as well as it is a keto acid. Now, after accepting the phosphoenol pyruvic acid with the bicarbonate ion, it will be formed into a 4th carbon compound, that is the oxaloacetic acid. So in case of C4 cycle, the first stable compound is the oxaloacetic acid. So what did I told you just? So in case of C4 plants, first of all, the whole process in case of C4 plants, the whole process is subdivided into two locations. One of them is the mesophyll cell chloroplast and the second one is the bundle-sheath chloroplast. And you have to remember that in case of both the chloroplast, the process is strictly limited or strictly restricted only to the stroma of the chloroplast. As all of you know that in case of light dependent reactions, it is occurring in the grana, and in case of light independent reaction, it will be occurring in the stroma. And as the C4 cycle is an example of the light independent process, it will always occur in the chloroplast stroma. So first of all, let's take these I have separated the board into two separate compartments. The upper one, I am representing the upper one segment is the mesophyll cell chloroplast. Mesophyll cell chloroplast, and obviously it will be stroma. And the lower portion of the board, I am representing it as a bundle-sheath. Bundle-sheath chloroplast. It will be also stroma. Now, first of all, the reaction will start from the mesophyll cell chloroplast. So, first of all, carbon dioxide will enter into the mesophyll cell chloroplast by what? By stomata. As you all know, there are basically different ways, or there are basically different structures that plants have by which gaseous transportation occur. One of them, the main component, is the stomata, second one is the lenticule, and the third one is the cuticle. But the majority of the gaseous exchange that basically takes place through the stomata, and it is basically surrounded by guard cell. You all know the structure of the stomata. So carbon dioxide will enter the mesophyll cell chloroplast through stomata, and it will react with water. It will react with water to form carbonic acid. You all know the formula of the carbonic acid, that is H2CO3, and the enzyme will be carbonic anhydrase. The enzyme that is the carbonic anhydrase, all of you have to remember that carbonic anhydrase is the fastest enzyme that any living organisms occupy. It is the most fastest enzyme. It can almost react as many as 10 to the power 6 molecules of carbon dioxide with water per second. So it will be the carbonic anhydrase. I am writing the short form of the carbonic anhydrase, that is the C.A, and the newly formed carbonic acid will be broken up into two fragments or broken up into two ions. One of them is the hydrogen ion and the second one is the bicarbonate ion. So bicarbonate and hydrogen ion, H plus and HCO3 minus. Now as you can see, the carbon containing ion is the bicarbonate ion. So the portion or the part of the carbonic acid which will be accepted by a second compound will be the bicarbonate ion. Here in case of mesophyll cell chloroplast, the primary acceptor of the bicarbonate ion is the what? Is the phosphoenol pyruvic acid. Phosphoenol pyruvic acid. Phosphoenol pyruvic acid is basically a 3-carbon compound. Now what is the difference between pyruvic acid and the phosphoenol pyruvic acid? It is just as simple as that, presence of phosphate and the absence of phosphate. In case of the pyruvic acid, the phosphate will not be present, but in case of the phosphoenol pyruvic acid, a phosphate group will be attached at the 3rd carbon of the pyruvic acid. As you all know that pyruvic acid is basically a 3-carbon acid. What kind of acid? It is a keto acid because it contains a ketone group. Now phosphoenol pyruvic acid will be reacting. Phosphoenol pyruvic acid will be reacting with the bicarbonate ion to form oxaloacetic acid, the first stable compound in case of C4 plants, and as it possesses 4 carbon in its structure, the whole process is known as the C4 cycle. So the first stable compound will be oxaloacetic acid, and it is a 4-carbon compound. 4-carbon compound, and the enzyme, the rate limiting enzyme, the principal rate limiting enzyme for this particular step is the PEP carboxylase, phosphoenol pyruvate carboxylase. The principal rate limiting enzyme is the PEP carboxylase. PEP carboxylase basically an enzyme which combines two components. One is the bicarbonate ion and second one is the phosphoenol pyruvic acid. Together it combining these two compounds to form a second or a 4-carbon compound, that is the oxaloacetic acid. Now this oxaloacetic acid will be oxidized. This oxaloacetic acid will be, this oxaloacetic acid will be reduced into another organic acid, that is the malic acid. As we all know that in case of oxidation reduction process, if there is oxidation of a compound, there must be reduction of another compound which will couple the whole process. As we all know that it is together called the redox potential. So there must be some electron acceptor and there must be some electron donor. Now as I am saying to all of you that oxaloacetic acid will be reduced, that means oxaloacetic acid will be accepting electrons and hydrogen, but there must be some donor of that particular electron and hydrogen. In case of light dependent phase, what we have seen? In case of light dependent phase, we have seen the production of the ATP, NADPH and oxygen. Oxygen is liberated from the stomata, but what remains in the plant is basically the ATP, and the NADPH, nicotinamide adenine dinucleotide phosphate hydrogen. So here in this case, the NADPH will be acting as a donor of electron and hydrogen, where it will donate its electron and hydrogen to the what? To the oxaloacetic acid, and ultimately reduce the latter and get itself oxidized. So now oxaloacetic acid will be reduced malic acid. Malic acid is also a 4-carbon compound, as there is no deduction of the carbon molecule from the oxaloacetic acid. Now there is a oxidation reduction process is occurring, that is NADPH, which is formed in the light dependent phase, that is the cyclic and the non-cyclic photophosphorylation, which have already made the videos. If you don't watch yet the videos of the cyclic and the non-cyclic photophosphorylation, please do have a watch. You will get a complete idea of the cyclic and the non-cyclic photophosphorylation. So come back to the point. Now NADPH will be oxidized into NADP. NADPH will be oxidized into NADP. That means it is donating a proton and the electron, and the proton and the electron will be accepted by the oxaloacetic acid to form a reduced substance, that is the malic acid. Now this malic acid is the ultimate product that is formed in the mesophyll cell chloroplast. Now this mesophyll cell chloroplast, from this mesophyll cell chloroplast, the malic acid which is formed in the stromal portion of the chloroplast or the mesophyll cell will be transported into the bundle-sheath chloroplast. Here is the most important event that is occurring only in case of C4 plants. That is the fixation of the carbon dioxide will be occurring in 2 different stages. One of them that I have already discussed you, that is the mesophyll cell chloroplast. Now I am going to discuss you about the fixation or the processes that is involved in the bundle-sheath chloroplast. Now in case of bundle-sheath chloroplast, there is a basic, or there is the most important and a unique feature that we will get in the bundle-sheath chloroplast. What is it? It's the Kranz anatomy. Bundle-sheath chloroplast shows a unique feature. This unique feature is the Kranz anatomy. Write it down, students. It will be very important and you will be getting it in the Neet for 100% sure. You will be getting this question, the which plant or which name some plants which shows Kranz anatomy. It will be C4 plants, and in the end of the video I am going to explain, or I am going to give you some examples of the C4 plants that will be very, very important for your Neet examination. Now Kranz anatomy basically a cluster of cells that we generally get to see in the bundle cells, that is the xylem and the phloem. A cluster of cells that is surround the xylem and the phloem in a circle way. So this particular Kranz anatomy is very, very important characteristics of the bundle-sheath chloroplast that you can see in the C4 plants. Now there are connection between the different organelles or different cells of the tissues in the plants by plasmodesmata. As you all know that plasmodesmata is basically a gap junction in case of plant cells by which cells are connected to each other. Here also plasmodesmata will be connected. Plasmodesmata will be performing the connector between the bundle-sheath chloroplast and the mesophyll cell chloroplast. Here it is the mesophyll cell chloroplast stroma, and here is the bundle-sheath chloroplast. Now the reduced form of the malic acid that has been formed in the mesophyll cell chloroplast will be transported. Where? It will be transported in the bundle-sheath chloroplast through plasmodesmata. Now malic acid have already reached. the bundle-sheath chloroplast. Now what will happen? Now this malic acid will be again oxidized into something, as well as there will be a deduction of the carbon dioxide. So the whole process, you can call it oxidative decarboxylation. So there will be occurrence of oxidative decarboxylation of malic acid. That means the carbon number of the malic acid will be reduced. How many molecules of, by how many molecules of carbon? By a single number of carbon, as carbon dioxide contains a single molecule of carbon, as well as there is a next, or as well as a second event will be occurring with the malic acid. That is what? That is the oxidation of the malic acid. So now malic acid will be oxidatively decarboxylated. Write it down, students. It is very much important steps. Oxidative decarboxylation. That means oxidation and decarboxylation are together coupled to each other. Oxidation and decarboxylation are together coupled with each other. So first of all, we will be seeing what will be the processes of oxidation. Now malic acid will be oxidized by the help of, again, NADP. The NADP plus which is formed here will be again, the NADP plus will be again reduced to NADPH plus H plus. That means the NADP is accepting the electron. The NADP is accepting the electron from the malic acid, as well as a carbon dioxide will be, clipped off, will be released from the malic acid. So the molecule, or so the compound which will be formed after the oxidative decarboxylation will be containing a 3-carbon molecule, and the molecule is none other pyruvic acid. Pyruvic acid is a 3-carbon molecule which you will obtain after the oxidative decarboxylation of the malic acid. So the process, you have to write it down in your examination copy. Name the process, that is the oxidative decarboxylation. Oxidative decarboxylation. So after the oxidative decarboxylation, what will form? That is the pyruvic acid. Pyruvic acid is the 3-carbon compound that you can see. Pyruvic acid will be again transported back to the mesophyll cell chloroplast, where it will be phosphorylated at its 3rd carbon to form again phosphoenol pyruvic acid, and the whole process will be repeating once again. So the name C4 cycle will be justified. So the pyruvic acid that has formed, will be again transported back to the mesophyll cell chloroplast through plasmodesmata. Now after reaching the mesophyll cell chloroplast stroma through the plasmodesmata, there will be a phosphorylation of the pyruvic acid at the 3rd carbon by what? What is the phosphate donor here in this case? That is the ATP which has been formed in the light dependent phase. So the pyruvic acid will be phosphorylated. Pyruvic acid will be phosphorylated by ATP. ATP is the donor of the phosphate, and it will be phosphorylated to form phosphoenol pyruvic acid, and ATP will be changed into ADP. So as a result, you can see that the onset or the starting point of the process, that is the phosphoenol pyruvic acid, is again forming. So the whole name, that is the C4 cycle. Cycle means what? Cycle means the starting point and the ending point will be the same. So here in this case we can see that the whole process where it started, from where it is started? It is started from the acceptor, that is the phosphoenol pyruvic acid is again forming. So what are the basic questions is that you are getting from this particular topic? First one is the what is the first acceptor of carbon dioxide in case of C4 cycle? The answer will definitely be the phosphoenol pyruvic acid. Second question, what is the first stable compound that has been formed in the C4 cycle? It will be oxaloacetic acid, and as oxaloacetic acid contains 4 carbon in the structure, it is also called the C4 cycle. Now, the C4 cycle is also known as the Hatch and Slack cycle. The C4 cycle is also known as the Hatch and Slack. Hatch and Slack are the name of the two scientists, and those two scientists were basically experimented and discovered this whole process at first, and for their name it is also called Hatch and Slack cycle. So there is involvement now where in this whole process glucose is being formed. We can see that in the whole process glucose is never produced. Now where is glucose is being producing? The glucose is being producing in the bundle-sheath chloroplast. The carbon dioxide which is being liberated from the malic acid at the oxidative dephosphorylation stage will be entrapped by rubisco, and the principal acceptor of this particular carbon dioxide will be ribulose 1,5-biphosphate. So the carbon dioxide that has been released will be accepted by the RUBP. You all know that the full form of the RUBP, ribulose 1,5-biphosphate, and the RUBP will be forming the first stable product, that is the 3-PGA. That means 3-phosphoglyceric acid, and the 3-phosphoglyceric acid will be ultimately forming again a C3 cycle where you can get a glucose. Now if you haven't watched it, the C3 cycle, how plants perform C3 cycle or Kelvin cycle in order to fix carbon dioxide, please do have a watch my videos. I have already made the videos about the C3 processes, how plants perform C3 process and they fix the carbon dioxide in the chloroplast stroma. So do have a watch. Now after forming the 3-PGA, the 3-PGA in the different intermediate steps will be producing glucose. You all know that how C3 plants perform fixation of the carbon dioxide in the C3 processes. The first product is the 3-phosphoglyceric acid, then 1,3-biphosphoglyceric acid, then after 1,3-biphosphoglyceric acid there is formation of 3-phosphoglyceraldehyde, dihydroxyacetone phosphate. After that there are isomerization, so that's a different story. So here the carbon dioxide that has been liberated after the oxidative decarboxylation will be entered into the C3 cycle. So write it down, students. In case of the C4 plants, there is occurrence of both the cycle. One is the C3 cycle and second one is the C4 cycle. That means C4 plants can perform both the processes, that is the C3 as well as the C4. Now where does this C3 occurs? You have to remember it. The C3 process in case of C4 plants is being occurring within the bundle-sheath chloroplast, not within the mesophyll cell chloroplast. So we can see that in the C4 plants, the fixation of carbon dioxide into glucose is basically in the bundle-sheath chloroplast. But first of all, carbon dioxide is accepted by the compound, that is the phosphoenol pyruvic acid, which is present at the mesophyll cell chloroplast. So that's all about C4. That's all about C4 cycle. Now, what are the basic characteristics that we will get? What are the basic features or what are the basic characteristics that we will get or you can see in the C4 plant? First of all, C4 plants are very much resistant to the hot climate or aridity condition. They can sustain their life in very much hot climate. They will not die in the hot climate situation. First of all, then they have a very low carbon dioxide compensation point. That means C4 plants can perform photosynthesis if the atmospheric carbon dioxide level is very low. Third one, the C4 plants will be showing crunch anatomy in the bundle-sheath chloroplast. Fourth point, the rate of carbon dioxide fixation in the C4 plant is comparatively higher than the C4 plants, than the C3 plants, and C3 plants have two carbon dioxide fixating enzyme. First one is the PEP carboxylase and the second one is the Rubisco. Here carbon dioxide will be accepting by the RUBP by Rubisco. The full form of the Rubisco, ribulose bisphosphate carboxylase oxygenase. So what are the basic characteristics? That is, I am going to tell you again. First one, that C4 plants can sustain its life in very much hot climate where the environment or the temperature is above 30 degrees Celsius. Next, C4 plants show crunch anatomy at the bundle-sheath chloroplast. Third one, the carbon dioxide fixation point of the C4 plant is very much low. That means C4 plants can perform fixation of the carbon dioxide if there is a very little amount of carbon dioxide is present in the atmosphere. And the last one, that is, there is fixation of the carbon dioxide is being occurring by two different enzymes in two different locations. One of them is the bundle-sheath chloroplast by Rubisco, and the first one, that is the mesophyll cell chloroplast by PEP carboxylase. And what are the basic examples of C4 plants? The first and the most important examples of C4 plants are zeamase. Zeamase is one of the most suitable and most phenomenal plant that shows C4 processes. Second one is the sugarcane, and there are some also grasses which are also perform C4 cycles. So these are all the examples. First of all, you have to remember it. What are the examples? Sugarcane. Second one is the what I have told you, sugarcane, and next zeamase, and some of the grasses are the examples of the plants which perform C4 cycle. So this is all about C4 cycle. You will be getting questions in your exams from the C4 cycle, and it is very much important for the Neet examination. You will be getting so many questions, as like 3 to 4 questions are each and every year you will be getting in the Neet examination. So it's very much important. So do write the notes that we are getting from the videos, and if you like the videos, do like the videos, do share with your friends. If you haven't subscribed my channel, then do subscribe my channel, and don't forget to press the bell icon. Thank you.